Welcome to today's webinar, New Core Skills Standards for Bioscience Technicians, Making Them Work for Us. We are pleased to have today's moderator, Elaine Johnson, PI for BioLink from City College, San Francisco. Welcome, Elaine.
Hi, everyone. I am delighted that you're all here today. We've got a large number of participants talking about the new core skill standards for bioscience technicians and making them work for us. We acknowledge at this point also the huge contribution from the Community College Consortium for Bioscience Credentials, and the director of that entire consortium, funded by the Department of Labor, is Russ Reed, and he's on the call today. So if we have any questions for him, we can feel free to ask those. We're looking at this from the standpoint of the National Science Foundation funded ATE projects and centers, and one of those is BioLink, and I'm the director of BioLink. I'm based at City College of San Francisco, and BioLink works to increase the number and diversity of the well-educated technicians in the bioscience workforce. And our members are workforce-oriented college and high school programs, instructors, industry, students, and our website is bio-link.org. We have some wonderful presenters today that I know that many of you know, but if you don't, I know that you'll be delighted to hear what they have to say and to enter in, and this is our opportunity to ask questions from each other, to make comments, to share whatever we can in this time together, talking about the new skills standards for bioscience technicians. One of the presenters today is Dr. Jeanette Mowry. She is part of the BioLink leadership team and is from Madison College in Madison, Wisconsin. She has been one of the key players in preparing the lab skills and the core skills. And so as she presents, she's got huge expertise in this area. And that's terrific. The next presenter is Dr. Lisa Seidman, who is also at Madison College. She's a co-PI on the BioLink grant and has been since the beginning of BioLink, which was first funded in 1998. Lisa was one of the people who started the first biotech program in the whole United States. And in doing so, she made a major contribution to the materials available, the lab calculations, the lab manual, materials that have been used across the United States. And she is still one of the leads in the instructional materials clearinghouse for BioLink. I'm also interested in authentic assessment and how we might use these skills standards. So we are very happy that Lisa is presenting today. Another key player in this is Dr. Linda Rafus from Bucks County College in Pennsylvania. And she is the director of NBC Squared, which is the Northeast BioManufacturing Center and Collaborative. But even though that name infers that it's funded as a regional ATE center or Advanced Technological Education Center by the National Science Foundation, Uh, their group has produced a textbook and lab manual that's used across the nation. And she has been very active in the skill standards, and particularly from the biomanufacturing, but also in the core standards. And finally, we have Dr. Linnea Fletcher, who's the department chair for biotechnology at Austin Community College, but she's also the principal investigator for a relatively new AC squared Austin Community College uh, regional center, and it's a BioLink regional center. So that's an interesting way that we've been looking at our sustainability. Linnea is also a former program officer at the National Science Foundation. So as we begin, we'd also like to hear about you, and we'd ask you, what is your primary affiliation? If you will fill this out, we'll be able to see the results on the screen. And there's primarily the two-year community college representation, but some from 4 years, some high schools, a little bit of industry, and some other. Other might be national labs, nonprofits, things like that. So we'd be really interested in hearing more about you, and I have the list of attendees, so I will be able to follow up with you, but I also would love it if you could follow up with me if there's something of special interest so we can get back to you with materials. One of the things that happens in these webinars is that sometimes people get together in a room and there's more people that are actually participating than is shown by the number of participants. And sometimes people actually bring a whole room together and project these webinars. So we would like to know how many are participating today from the registration that you put in. Well, I guess that's it. We're all doing this on our own, so that gives us even more chance to want to interact with each other as we go on with this webinar that I will now turn over to Dr. Lisa Seidman.
Okay, thanks so much, Elaine. And I'm going to begin a little by going through the agenda very briefly. We're going to begin with an introduction about a recent report that came out about reproducibility in science, which is a contemporary hot topic. And as you will see, although this may seem like a little bit of a diversion, it is directly related to the core skill standards. And after that introduction, Jeanette Mowry will be talking about the new core skill standards, how they came to be, a little bit about them, their background. We'll come back a bit to this issue of reproducibility, and then we're going to launch into teaching resources, beginning with those resources that are available from BioLink, and then turning the microphone over to Dr. Linda Rafus, who will be talking to us about the NBC squared resources. And then at the very end, we're going to have a very interesting case study about how Texas and how people in Texas have already been incorporating these skill standards into the work that they're doing. So, by way of introduction, this is a headline, a real one, that came out on January 18th, a couple weeks ago, 2017. And in this headline it says, Cancer Reproducibility Project releases its first results. An open science effort to replicate dozens of cancer biology studies is off to a confusing start. Another headline we have from Science magazine, also January 18th, rigorous replication effort succeeds for just 2 out of 5 cancer papers. Going on from Science a couple days later, headline says mixed results from cancer replications unsettled field. And finally, BioPharm International has quite a lot to say about it. Reproducibility project only partially able to validate findings of prominent cancer studies. So what is this all about? Well, In 2012, Amgen researchers reported that they had failed to replicate 47 of 53 landmark cancer papers, which really concerned people leading up to, in 2013, the Reproducibility Project in cancer biology specifically. And this team of researchers just reported on January 19, 2017, the results from 5 studies, where they were successful in replicating 2 out of the 5 studies. 1 study was clearly not replicated, and 2 studies were inconclusive. So whether the glass is half full or half empty, people are concerned about this. And why is this a big deal? Well, first of all, there are consequences for patients, treatments, prevention. So obviously every citizen is concerned about not being able to replicate very important cancer research. But the pharmaceutical industry and the biotechnology industry is very concerned about the huge financial implications, because we know that many drugs, many treatments are being developed, millions of dollars, billions of dollars are going into developing treatments and interventions that are based on understanding perhaps a cancer gene or a cellular receptor or a ligand or some kind of fundamental understanding of how cancer works. And so if these treatments are being based on faulty information, it could be people's careers, years, and all sorts of resources are being wasted. This kind of thing also affects the public's view of science, and I suppose in a really pessimistic way we might say it might make the public a little bit more open to, say, alternate facts. And finally, this kind of result strikes at the heart of what science is all about, because science is about uncovering the nature of the universe, uncovering truths about nature, and these truths should be replicable wherever we are, any place, as long as we have the right resources and the right knowledge, we should be able to reproduce them. And so this is a pretty scary kind of result. So journalists and Science, Nature, they've already responded to this by adding much more detail in research reports. I've seen this already in Science magazine, for example. And they've used the term transparency. And by this they mean they have increased substantially the documentation of materials and methods. And they also use the word transparency as we might use the word traceability, encouraging people to look back at their raw materials and at all of the resources that went into doing a research paper. And I'm going to then come back to this in a little while. But before I do, Jeanette Mowry is going to talk to you about the core skill standards and where they came from and how they were developed. Jeanette.
Okay, we have a little microphone changeover here. So before I start talking about the process for developing the core skill standards, I want to make it clear that this booklet has now been published. It's a 30-page booklet. It's available electronically and in paper form, and I will show a slide later on about how you can access that. And one of the easiest ways is also just to email me about it. So a very key point here, though, is these core skill standards are not by any means the first skill standards in bioscience and biotechnology. Early efforts began in the early 90s with agricultural biotechnology skill standards and with EDC skill standards for biotechnology. They were followed by some skill standards for biomanufacturing, some skill standards by the Washington State skill standards that dealt with not only research and development, but biomanufacturing and regulatory affairs. Those skill standards were last developed in 2007. And then there were some efforts by the Department of Labor to build a pyramid of skill standards, and that's been published in 2008. So this is not by any means a new idea to come up with skill standards, but what is unique about this effort is that it's focusing on the core, what is in common across industry subsectors. So this particular effort comes out of a Department of Labor TACT grant, a round 2 TACT grant, and this was awarded to Forsyth Technical College. Russ Reed is the director, or was the director. It went on for 4 years, ending just last year. TACT stands for Trade Adjustment Assistance Community College and Career Training Program, hence the reason to have the acronym. Nobody wants to say that very often. And this grant had a number of objectives, but one of the key deliverables of this grant was to identify these core bioscience skill standards. This particular Department of Labor grant was named the Community College Consortium for Bioscience Credentials, and you can see in the upper right-hand corner the logo for that particular organization, C3BC, and it's on many of our slides. And that might be important later if you try to access some of the other materials of the grant, that you know what the grant name was. So I need to tell you a little bit about the organization of the grant, because that impacted what we did in terms of core skill standards. So the consortium was organized, the 12 College Consortium, led by Russ Reed, was organized into 4 hubs. And I'm not going to talk about the Learning Technologies Hub, even though it was very important to some of the work that different colleges were doing around the country. But I'm going to focus instead on the 3 hubs that dealt with industry subsectors. And these hubs were people that you are familiar with are leading those hubs. The Bioscience Lab Skills Hub was led by Dr. Elaine Johnson. The BioManufacturing Hub was led by Sonia Wallman of the Northeast BioManufacturing Center, and now that center is now co-led by Linda Rafus and Maggie Byrons. And then there was a Medical Device Hub, which was led by Dr. Sung-Young Lee. So 2 of these hubs were actually represented by ATE centers already. So each of these hubs had their own skill standards, at least the first 2 did, and then the Medical Devices Hub developed their skill standards over the lifetime of the grant. So as I said, there were other objectives for this grant: improving and expanding recruitment, accelerating completion time and credentialing programs, building capacity for bioscience education, all with the goal of really assisting displaced workers. And the key area that we focused on was to harmonize a set of bioscience core skills. What does harmonization mean? It simply means that of the 3 industry subsectors and their skill standards, what are the skills that are held in common across all of those 3 sectors? Not that it's a comprehensive effort, it's simply an effort to determine what is in common. Why would we want to do this? Why would we want to focus on core skill standards? Well, for a variety of reasons, but I think the primary driver for us was the tremendous diversity of entry-level technician positions. Those of us who have been teaching students to prepare them for these jobs for years know how much diversity there is in those positions. And it can become challenging to find out what's important when you have limited time. What do you focus your teaching on? And all skill standards have the value of having students have a vocabulary so that they know what they know and they can articulate what they know in a way that industry supervisors can understand. And also that these skill standards can really be a bonus to help educators develop curriculum and courses. They also provide a means for measuring student performance if they're used in that way. And a sort of a bonus I found in this particular grant was that it really facilitates the process to award credit for prior learning, because these skills that were identified as being core, in many cases, don't really scream bio at you. They really have a lot in common with other manufacturing industries. And that allows students to see how they might have prior experience in another industry and could come to work in biotechnology or vice versa. And as Dr. Linnea Fletcher will talk about later in this webinar, the possibility exists to develop a credential based on those core competencies. We've got this silly arrow that I keep moving around here. So I need to tell you a little bit about the process, and in order to do that, I have to tell you a little bit about my team that worked on this. So for each of those industry subsectors, the consortium chose a representative to meet and determine what was core. So for biomanufacturing subsector, the representative was Linda Rafus, who you'll hear from later in the webinar. And from lab skills, that representative was myself. And from medical devices, that representative was Vivian on Windward, who also has a lot of experience in the biomanufacturing arena. So one thing I forgot to mention in an earlier slide, this title of laboratory skills as an industry subsector. It's not really an industry subsector as we know it, but when we originally conceived of this particular hub, it was labeled research and development. And many of us who teach students to go and work in research and development labs also teach those same students the skills they need to go into small-scale production and testing laboratories. So the skills and the scale of the bench lab scale that they work on is very similar. So we needed a bigger umbrella term, and laboratory skills is it. It's not perfect, but it's what we've got. So what we did was we looked at the list of tasks from each of the 3 hubs, and we looked for areas of commonality. And we formed many matrices like this. We were pretty comprehensive. We had multiple meetings. And then where there were common elements, we worked on figuring out language that would work for all 3 subsectors. So that's what's shown in this final column, is the idea that we would develop language that would work for all 3 of the different subsectors. And that was sometimes challenging, sometimes not so much, but it was still a big part of what we worked on. And this is only showing, for simplicity's sake, just showing a few of those ideas. So, oh, I went the wrong way. So what kind of format should we use for these skill standards? And there's a variety of formats used in the past, anything from a simple list of tasks to much more complicated formats. But the one that we ended up choosing was one that was used by the Washington State Skill Standards, and their latest iteration was in 2008. And it involves choosing a pretty large work function called a critical work function that is a major responsibility of the job. And there would be, presumably not that many of those critical work functions. And then within each critical work function, there would be key activities, and there are multiple key activities to accomplish a single critical work function. And then performance indicators would be specific ways that we would know that the individual is doing this activity correctly. And of course, underlying technical knowledge and skill, and an assessment to determine proficiency. So that was the format that we chose, and that's what these final core skill standards are in. So of course, oh, and I should also say that after the process of the team figuring out the first draft of common language for the core, it was vetted by numerous educators over the course of the grant and multiple meetings at BioLink and NBC2 and others to get educator feedback. So we weren't the only educators that were involved in it. But the industry input and recognition process is really 3-tiered. And the first tier, I'm not going to neglect our core team, because all of us had worked in industry and had combined more than 50 years of teaching in biotechnology workforce programs that answer to industry advisory boards, placing interns, talking to internship supervisors about what students know and what we would like for them to know. And so there was quite a bit of experience just in that core team. And then our C3BC consortium had face-to-face meetings throughout the life of the grant, and we had a national advisory council with industry members that was very helpful in giving us feedback as we went along. And then the final push for that was an online survey where we reached out to industry supervisors of entry-level technicians. And in that survey, we had input from 22 industry supervisors across the 3 industry subsectors to give us feedback on it. So I need to make sure that people don't walk away with the misconception that these are the bioscience skill standards. They are not. They are the core bioscience skill standards. They are necessary, but they are in no way sufficient. There are skills that are necessary in all those other subsectors that are not in the core. So just keep that in mind. If you look at them, you'll see some of the missing things. They'll come out, they'll scream out at you, especially if you're in lab skills biomanufacturing. There won't be as much wet lab as you would like, etc. But that's because we focused on this core. Now, where can we find these core skill standards? They've been posted multiple places, the BioLink website, of course. The National Center for the Biotechnology Workforce has them front and center on their homepage. You have to scroll down a little bit, but you can click right on that and get the booklet downloaded. And other materials from the C3BC Consortium grant are posted at skillscommons.org as well. So you can get the skill standards there, but you can also get the courses, syllabi, a lot of other material that were deliverables from that grant you do have to, because skillscommons.org is the repository for all the materials from Department of Labor tax grants and maybe all other grants too, I don't really know. You have to know the name of the grant. So Community College Consortium for Bioscience Credentials is important to be able to navigate that. But if you're curious and wanted to go look for other materials there, that's what you would need. And as always, you can email us for electronic or paper copies. There's my email, and it won't be too hard to find me from the BioLink website if you forget my email. So with that, I'm going to turn it back over to Lisa.
Okay, and I'm going to return to this issue of reproducibility. So I would say we're probably pretty convinced that reproducibility is fundamental to laboratory research and, in fact, to any kind of laboratory testing, whether it's clinical or environmental or whatever. But I think it should also be clear that reproducibility and reducing variation is absolutely essential if one is in any kind of production, including biomanufacturing or medical devices. And in fact, reducing variability is a huge part of any quality system in any production setting. So if we say reproducibility or reducing variability, increasing reproducibility and reducing variability are critical, then we can see that it's not going to be a big surprise that these are found in the core skill standards. So in fact, the core skill standards do very much direct the issues of reproducibility and reducing variability. And in fact, I think that one could argue that even though medical devices and laboratory and biomanufacturing in some ways are quite disparate, in fact, there is a unifying theme that they all do hold in common, perhaps a core of the core. And that core of the core is to address reproducibility and reduce variability. And so with that idea in mind and looking for that unifying principle, we can just take a quick look at some of the critical work functions that are found in the core skill standards. So as Jeanette mentioned, they're divided up into these buckets, these big buckets. And the big buckets are provide routine facility support, perform measurements, tests, and assays, comply with applicable regulations and standards, manage and communicate information, perform mathematical manipulations, maintain a safe and productive work environment. In the interest of time, we are certainly not going to look at every page of the core skill standards and show you every single core critical work function, but just a couple of examples. Oops, wrong arrow. Come down here. So here is a critical work function to provide routine facility support. And if we look at this, then we can see that, for example, there is proper documentation practices and preventive maintenance is performed in an equipment logbook. So if we think about, again, the issue of transparency, which the journalist mentioned, that would be the same thing basically as documentation. And we see this. Documentation is found as a key thing throughout the core skill standards. Preventative maintenance is performed, and again, this is exactly the sort of thing that is going to ensure consistency in any kind of environment, whether it's laboratory or manufacturing. We also see other issues that are very much key for maintaining consistency, like calibration, verification, qualification and validation, general principles of metrology, critical for maintaining low variability, high reproducibility. And so we see that in this particular critical work function. We see it again. Here's another one. Perform measurements, tests, and assays, another critical work function. And again, we see these same issues coming up. For example, types and causes of measurement error, distinguishing between accuracy and precision. All of these are tools. All of these are ways in which we aim to reduce variability in any kind of environment. And again, we see the importance of documentation coming up over and over and over again. And measurements and tests and measurement tools used correctly again. If we look at another one of the critical work functions, comply with applicable regulations and standards. Now, that may not be so immediately apparent how that relates to variability and reproducibility, but if one thinks about, for example, the FDA, that is one of the major regulators in our profession, FDA says that products must be safe, reliable, and effective, where that reliability depends on having low variability, high reproducibility. And so again, we see throughout regulations and standards an emphasis on the things that would give you low variability, high reproducibility, like handling deviations appropriately, document control, change control. More about regulations and standards. Here we would see traceability. And as I mentioned previously, this whole idea of transparency that the journalists, the term they use, very tied to the idea of traceability, which is where you can identify the raw materials, the reagents, everything that went into making a product. Very important in terms of reproducibility. And again, more in the applicable regulations and standards having to do with the quality system, understanding change control, and so on. So we mentioned before, what is required to reduce variability and promote reproducibility? Well, the journalist solution, not surprisingly, is to print something. Well, they're journalists, and so they print details, and they've done a great job at that. But we're educators, and so, not surprisingly, our solution has to do with education. And so I think we have maybe a responsibility here to work as educators in this arena. And as a scientist, I guess, I want to give you some data to consider to prove my assertion. So I'm going to talk to you about 1 molar tris buffer pH 8. For about 10 years or so in our program, we have been educating in what we call PostBack Training from PostBack Education Program. We've been educating people who already have a bachelor's degree and in many cases who have experience working as technicians, but who want to get more into biotechnology. And we also have a few students in this cohort who are seniors in a 4-year program, but already have all their chemistry and most of their biology courses. So we've been educating people who have a very high level of expertise, certainly high enough to be entry-level technicians somewhere, certainly entry-level researchers or entry-level people or even more advanced people in many workplaces. One of the activities we always ask them to do every year at the beginning of the semester is to make 1 molar tris buffer pH 8. And here's some data from them that they put up on the board. In the left-hand column, you can see the initials. Those would be the teams or the individual people who made each reagent. The pH that each of those teams achieved, and if you look down that middle column, you will see that their pHs are pretty consistent. We asked them to achieve 8. They get 8.04, 8.06, 8.00, 8.02, and so on. That looks pretty good. But if you dig a little bit deeper and you ask them to check the conductivity of their solutions, what you see is that the consistency is not so great. 31.1 millisiemens per centimeter, 19.9, 33.1, 26.8, and so on. Now, no matter how you cut this, at least one of these teams is not reproducible with the others. Hard to say who's right and who's wrong, but we know that at least one of the teams is, in fact, different. And if, as educators, we say to the students, take a look at your data, take a look at what you did, go back through your documentation, talk to one another, in almost all cases, we can pretty quickly have the students figure out why one group is different than the others. You can address this kind of irreproducibility or variability simply by asking them to think about it, by using an educator's approach. And so, what does this mean? Well, first of all, it obviously means that we cannot assume that individuals have the core skills that are in the core skill standards, even if they have a bachelor's degree or even if they have some experience. I think second of all, we can say that education has a critical role here and that we can address these core skill standards explicitly in our classes. And in fact, one could even argue that maybe we are in a special position to do so, and maybe we have a special responsibility to contribute our knowledge and our expertise to this discussion of reproducibility. And so with that, I'm going to take us to questions. And I think I'll turn the mic back over to Elaine, who will help us with the questions part.
Elaine, do we see any questions? Well, I guess we will just keep going then. So the next thing we're going to talk about is resources, and this is really from the point of view of if I was a relatively new teacher and had no idea how I wanted to address these skill standards, where would I go for help? And fortunately, because some of us have been teaching in this field for a long time, there are a number of resources available to you. And so I'll talk about three main groups: the BioLink resources, and then the NC Bio Network resources, and then Linda Rafus will talk about biomanufacturing resources on biomanufacturing.org. Many of you are already familiar with the Courses in a Box section of the BioLink website, but a number of those courses really do address the area of reproducibility and the skill standards right away. So we can go right to the list of those courses in a box and see which ones might be applicable. The Quality Regulations and Standards course in a box actually consists of two courses, one from Salt Lake Community College and one from Austin Community College. But there's a wealth of material that deal with the regulations and teaching students about how to work in a quality and regulated field. There's a laboratory math for biotechnology, which deals directly with the critical work function dealing with calculations. And there is a basic lab methods for in a regulated environment. One of our oldest courses that Lisa was instrumental in developing here at MATC, and she has books that support it. And that course has the most, probably, of any of the courses I mentioned, the most materials up on the website that could be applicable for learning about reproducibility. One more course that has to do with another one of the critical work functions, which is the hazardous materials and safety, which is not specifically addressing reproducibility, but certainly important in its own right. So I'll show you just one example course, which is the basic laboratory methods in a regulated environment, to give you an idea of what's there. I'm not able to move that arrow, so I'll just talk. This consists of a paragraph that describes the course, and all of these courses in a box have that sort of introductory paragraph. And then there's a list of any other published material that might be important in teaching the class. And then there's some material that's available without a login, and then there's an additional material section which does require a login. It's really easy to get, but that's because there are test banks and things that are specific for teachers, teacher notes in that section, and so we have kept that behind a password. So just to point out, again, I'm missing a point. Oh, here again, I've got the pointer back again. So in the additional materials section of this course, you can see there's a lot of activities around quality and some case studies around the quality and regulatory area. There's documentation on metrology, including weighing, volume, pH, and then a whole 3-unit section on solution making. All of that extremely important in the reproducibility area. And so aside from courses in a box, there is a new and really exciting resource that we've developed at BioLink in conjunction with Pellet Productions. Pellet Productions is the same video production company that does ATE TV, for those of you who are familiar with that. And this is an interactive movie or an interactive video production, depending on how you want to speak of it. And we did a webinar on this in our last webinar series a couple of months ago. But this is a great resource for teaching students about what it's like to work in biomanufacturing and in a regulated field. And the rationale for developing this this was an NSF grant-funded project, was because of how difficult it is to give students the flavor for what it's like to work in these areas while you're in a teaching lab in your institution. So in this interactive movie, students can play the role of one of 3 characters, or they can, by the time they finish, they can play all 3 characters. The 3 characters are Corinne Lawton, an upstream production technician, Kevin Turner, a QC analyst, and Asim Kamani, an upstream supervisor, an upstream production supervisor. And so the video people call this a choose your own adventure series, but that was not a term that we were familiar with, but we loved the idea of it because students can see the dilemmas that these technicians are faced with, and then they can make choices and then see how those choices play out in the movie. So that's actually filmed. It was really a blast to participate in this project. So an important resource, and it's at franklinbiologics.org. Right now, all of the video and interaction is available to you. In another couple of months, we will have a participant facilitator guide that will be there, which will give you a little bit more written material to use it in your classes. So, I want to talk about the NC BioNetwork too. I would be remiss if I didn't point out another really important place for resources, and that's the North Carolina ncbionetwork.org. And they have an amazing array of educational resources, mostly interactive e-learning tools, videos of things that are often difficult to communicate to students. And a number of those deal with the equipment that is needed to ensure reproducibility and with working in a regulated environment. So with that, I'm going to turn it, there's questions. I don't know that there will be another chance to ask questions after Linda Rafis talks about another resource. So I think that might be a good place to pause there.
There are some questions now.
Okay.
Let's
Okay.
type them in, and we want to make sure that we keep you involved in this as these presentations go on. So if you do have a question, please ask it and type it into the chat box. I've got somebody typing right now.
Okay.
So
we'll hang in there.
As soon as we get it. Okay. I know that there's been a lot of action on the chat box about who is who, and especially from the high school point of view, those people that are high school teachers and wanting to know how to get involved with BioLink. So I'm very happy about that, and I think that we have answered some of that, and certainly will continue to do so as we can have other presenters in this webinar. I have I'm waiting for the question. There we go. The question is, do you have any suggestions for credentialing of students? How will employers know that the students have a skill set?
Well,
we're going to have some comments later in this webinar about credentialing, but go ahead and respond if you have a response now.
Well, this is Jeanette still talking. This was is an ongoing issue. It's a huge issue because you can argue for and against something really formal. At a minimum, though, it gives you that vocabulary that the students can use to talk to their potential employer, and it gives an educator that vocabulary to be able to talk directly with employers about what they need. So, I mean, as far as a credential, I know that Linda Rafis is experimenting a little bit with this at the college level, and Linnea Fletcher has some experience with this at the high school level. So I'm going to defer that. They'll take that question when they come on board.
I'd like to add to that there is, within the state of California, there's a California sector of the biotech that's funded by the state, and currently they're offering some quality courses through one of the community colleges in preparation for industry certification through ASQ. And anybody could take that, and I think that it could be very fascinating to have our community college faculty, but also our high school teachers have that industry credential that is very meaningful to the industry. So we could talk more about that, but that is going to be a topic for one of the next webinars. We got one more person typing right now, and then we'll move on. And we'll address some of these as we, we've got one person saying that BASE is only for high school students. We are using this to give high school students college credit. And Linnea has indicated, as you can see in the chat box, that we're also promoting a Associate of Science and Applied Associate of Science students to do this as well. So let's move on, and then we can come back to more questions as we do this. So now we're going to move to Linda Rafus.
Hello, everybody. It's a pleasure to be here today. And just to add to that question that was asked before I begin my remarks, that yes, indeed, in the BioManufacturing Center, our grant is working on an exit exam, if you will, for those in a college biomanufacturing program. And it will be available to educators to give to graduates of biomanufacturing programs in order for, to address just that question: what kind of skills do students have when they graduate? So feel free to contact us about, our development and rollout of our exam. So as all of my colleagues have said, I'm here to represent the biomanufacturing sector, and our grant, the NBC2, that Elaine introduced in the beginning. This is a screenshot of our front of our web page. You can see there that we offer various professional development activities for instructors in the form of mini bioman, specialty topics that are 3 days that primarily are attended by community college faculty, and Protein is a Cash, which is our high school teacher workshop, where we teach aspects of biomanufacturing and some basic biotechnology to high school teachers that then they can use in their courses. And so please visit our website, www.biomanufacturing.org. And if moving ahead here, when you click on the curriculum tab of our website, you'll see right away all of the information that has to do with our textbook. So you see there all of our program units, and we're kind of divided along 10 basic jobs that have to do with biomanufacturing, such as facility, metrology, validation, and all of the topics you see there. And we also have many standard operating procedures, SOPs. We have other instructor resources like PowerPoint productions to go along with our curriculum. And as I go through this, I do want to point out that our curriculum is available on our website for free. You can custom choose the chapters and the SOPs and the ancillary materials that you want to use in your coursework as part of a whole course, as part of a module, however you see fit that will work in your course or program. So I'm going to be showing you a few of the activities that we do and materials that we have as they relate to the skill standards, but this whole set is available, and we invite you to look at our website. Just briefly about our center. So again, we want to coordinate local and regional efforts into a biomanufacturing education and training system to promote, create, and sustain a workforce, and to be the nationally recognized center of excellence that develops a world-class sustainable biomanufacturing workforce to improve the quality of life. Our leadership team, so Dr. Maggie Bryans is the PI and the lead, and I'm the co-lead with her at Montgomery County Community College. We're in neighboring counties, so we're fortunate that the two of us can work together, often meet face to face and work on a lot of the grant activities together. But we also have hubs in 4 other states: Finger Lakes Community College in New York, Professor Jim Hewlett's a hub leader there, Mike Fino at Maricosta in California, Dr. Bill Woodruff at Alamance in North Carolina, and Dr. Sen-Young Lee in Ivy Tech in Indiana. So I invite you also, depending on where you live, those of you on the webinar or whoever may listen to it after, to contact certainly Maggie or myself or any of the other co-PIs in their hubs around the country. So this is just a sample of a cover of our Introduction to Biomanufacturing textbook, but as I mentioned before, all of the materials are available online, and you can order custom manuals or print them yourself, but they will be available to your students. We also have, just again to show you, we have the BioManufacturing Lab Manual, and this is one again that's totally customizable with standard operating procedures and ancillary materials to teach all of our laboratory skills that go along with biomanufacturing, and also the core skill standards that you'll be able to find whatever you need as part of your ability, your desire to teach the subject of biomanufacturing. So I would like to go through a few of the critical work functions, as was talked about earlier by Lisa and Jeanette. That here's one, for example, that's perform measurements, tests, and assays. So for example, sampling procedures, chain of custody, samples of sufficient quality and quantity are taken. So if you take that particular critical work function, then for example, we have one, this is a specific standard operating procedure for microbial identification. If you are familiar with the biomanufacturing sector, you know that to make sure that your product meets predetermined quality and purity standards, they have to be set ahead of time, and then these tests are performed throughout the biomanufacturing run. So this is one, an available protocol that you can use to identify if there's any microbial contaminations in your biomanufacturing product. And what's nice about this is, one thing that I'd like to mention here is that based on every budget and need, we have throughout the sector, really throughout biomanufacturing, but also in the other sectors, we have procedures and that you can follow for every budget need. We recognize that some programs maybe have more startup funds or equipment or supply budgets, and others not as much. These API tests, for instance, are only about $6 a test, so you can buy them, and this is an activity that my students really, really love, to be able to figure out what are the specific microbial or microorganisms that are present in our samples. So that's one example of how we address that critical work function. Another example is participation in validation activities. So many of you might participate in validating, say, a piece of equipment. This is often one that we do in courses we teach at Bucks and at other of the colleges that are in the grant, and also across the country. For example, to the installation qualification, which is IQ, or the operational qualification OQ, or the performance qualification PQ. So we often use an autoclave because we have one available at Bucks, as I know they do at Montgomery County Community College and some of our other hub schools. But it could also be written so that you have any piece of equipment that you have, an IQ, OQ, or PQ, could be performed. So if we go to the page here, so for example, we have here the autoclave IQ protocol form, the autoclave monitoring form. And once again, of course, this is just a screenshot of one particular page of the website. But if you were to scroll down on this page, you would find the standard operating procedures for OQ, as well as for OQ of a particular of an autoclave, and also possibly performance of PQ. So that's really nice because it's all there. So under validation, you'll find the chapter, you'll find the standard operating procedures, you'll find other activities and related content. So everything that you need to know, again, if you scroll down the page, is available for you under validation. So this is just one part of the validation materials that we have. And then, for example, it wasn't there on the previous slide, but here's a protocol for the operational qualification of an autoclave. So the students can run through this and and then you can use it to do the OQ protocol for your autoclave. And again, if we can find a way to help you with some of these for other instruments you might have, this is, you know, we certainly are willing to work with you across the board. And then documentation. So I know that that came up earlier in Lisa and Jeanette's presentations. We know that you know, if you didn't write it down, the adage that if you didn't write it down, it didn't get done. So we have here, for example, a quality assurance. It's all very important to write everything down. We have, you know, our PowerPoint on quality, and we have other activities related to quality. But once again, if you see at the bottom of the page there, an exercise called CGMP popcorn exercise. So what's a CGMP popcorn exercise? Well, first of all, if you have a bag of unpopped popcorn and a microwave, you can do this laboratory. It's very engaging for the students. They, from receiving the unpopped bag of popcorn to the very end, determining if their popped bag of popcorn met the pre-specified conditions, you can teach a lot about current good manufacturing practices. So this is just a little bit of the top of the first page kind of explanation of the CGMP exercise. So the idea is you make a batch within a specified time frame, and it says 1.5 hours. I do find that I sometimes will do this over a couple of class periods because so that the students can become familiar with the documentation. So they work in teams. Some are in materials, some are in quality, some both quality assurance and quality control, some are in production actually making the popcorn, and then you go back to actually the final product. Did it meet the pre-specified specifications? So you understand the complexities of GMP, you gain appreciation of teamwork and cooperation in departments. Sometimes you feel frustrated, you feel rushed, you feel like, what's that group holding my stuff up over there? The students are saying that, they're going over there, hey, you're taking too long to get us our, you know, materials. So it makes for a very engaging, fun, and learning experience. And I know that, you know, many of the students who, you know, were involved in this exercise love it. And then they have to do all the documentation from writing the SOP, filling in the batch work, getting it signed and countersigned by management. And I think is Linnea just wrote in the chat that it's very cheap. Like I said earlier, if you're just joining, that if you have a, you know, you can print some pages, you have a microwave, and you have a bag of unpopped popcorn, you can do this activity. So we have a range of activities that meet the skill standards that can meet every budget. So I believe that was my last slide. So yes, so I will go back, and I encourage any of you to please contact us, you know, our PI, Dr. Maggie Bryans, myself, or any of the hub directors. And also please visit our website so that you can, you know, figure out what we have to offer. And since there was an interest, as we develop the, you know, and roll out the certification exam, we'll certainly give you more information on that as well on our website, that you know, that hopefully we'll have that available to instructors on a portal for instructors in the near future. So thank you very much for taking the time, you know, to listen to me and today. Thank you.
Thank you, Linda. And now we're going to turn this over to Linnea Fletcher.
Hi, can everyone hear me? I didn't get to test my.
You sound great.
Good. I didn't get to test my microphone at the beginning. So I just wanted to mention it's a great way to compare brands of popcorn. And then they understand how you do quality testing on popcorn. Anyway, I'm Linnea Fletcher, and I'm the AC Squared BioLink Regional Center Director. And thank you for participating on the webinar. I wanted to tell you a little bit about our regional center. We were funded about 2 years ago. It covers Texas and Kentucky, and some of the things that we're working on are the entry-level certificate for both placing it in high schools, and therefore we will talk a little bit about the certification exam that we have adopted. And of course, there'll be more about that in later webinars. We also are going to talk, we are working on contract services for companies. In fact, our wet lab incubator just opened up at the community college, where we have startup companies and students working on projects for startup companies. We work on using undergraduate research in biology courses that feed into biotech as a way to recruit students into the program. And so we're using biotech undergraduate research projects in these biology courses to hook students. We also are working on a variety of different types of statewide articulations in both Kentucky and Texas. And the other thing is we helped fund a survey. It's a national survey. BioLink was part of this also, that looked at what are some of the emerging trends in technician education. And interestingly, the survey that we funded in Texas mirrored that national survey, and all of this can be found at our website. So. Next, let's see here. Here we go. Didn't find the button right away. Oops. So I'm having a little problem advancing the slides. Can everyone still hear me? Hello? Uh-oh.
Sure, we can hear you fine, Linnea, and I'll be happy to advance the slides.
That would be good. I'm not sure why it's not doing that for me, but that would be fantastic.
So we're on the next slide now.
Okay. You are? I'm not.
Yeah.
That's interesting. So even though I can't see the next slide, I know what the next slide is about. So essentially, it's a picture of our program site at Austin Community College. So if you wanted to know, that's where the headquarters is for the Regional Center Grant. So could you advance slide one more to the one about the core standards being implemented in Texas?
I'm now on a slide that says a case study, news from Texas, level 1, level 2.
Great.
2 years or advanced technical. Okay.
Great. I can't see it, but I did produce it, so I know what it says. So one thing that was interesting for us in Texas, since we'd already been using skill standards, and when they started developing the intersection between medical devices and biomanufacturing, and then I call it R&D technician or lab skills, which define the core skills. In Texas, we decided to use that opportunity to align them or see if they were essentially what a level 1 certificate would be in biotechnology, with the idea that if that is the case, then we could create level 1 certificates using the core skills, and then from that also develop other programs. So we took that opportunity to create a level 2 certificate in biomanufacturing that comes off of the core skills. We also have our AAS degree, and we offer an advanced technical certificate, which is for baccalaureate students, and that also it comes off the level 1 and the core skills. And then finally, we are working on developing a medical device program that was to come off the core skills. Next slide.
We're on the next slide with jobs for 4-year students.
Things don't always work out. Jobs for for your students. Let's see. I wonder. So one thing we are doing too is, let's see. I was seeing if I could get that to advance. So one thing also is we did put the level 1 certificate also into 4-year programs. This is also helping us to develop articulations, because then students who are at 4-year schools can get a level 1, and at the same time we develop reverse articulations with these programs. Why don't we go ahead to the next slide?
This is the one on biotility.
Okay, good. Actually, if you go back to that other one, I think the other one indicated that at our school, the medical device program is not going to be coming off the core. It's going to be actually in a different area in advanced manufacturing. But if we go ahead, let's go ahead and go to biotility, because I think there was a lot of interest in the audience. And I would actually be interested to know how many people who are participating are using this certification exam. Is there any way to ask people this?
I'll type that in, and then people can answer yes.
Yeah. Because I see my chat box isn't working quite right either. So as part of the level 1 certificate that we were putting into high schools, we did look around the country or for any kind of certification exam, and that's when we discovered biotility. And I know that they had funding from NSF to start working on this, and then they had additional funding from the state of Florida, because like so many of the CTE programs in states, they're going the route that if there's any kind of workforce education, they prefer programs that end in a certification. That's why we're doing a level 1 in biotech in Texas, and they would like to have some sort of third-party certification exam. And at this point, this is the only one for the level 1 certificate. Now, there will be a webinar essentially with the developers of this certification exam, and I believe that's going to be the next webinar. One thing about it, in Texas, like many other states, CTE has adopted it with the help of some of our high school teachers like Jennifer Lazar, so that the $150 cost for the exam is now paid for by the state of Texas. So high school students don't have to pay for it. Okay. I think that's my presentation. This is what we've done so far with the core skill standards, and I will hand it back to Elaine.
Thanks, Linnea. It's so fascinating to hear what each region is doing, and I guess my comment on this is that the group of people makes such a difference. We've got the material, we've got ongoing materials, we've got ongoing connections, we have industry participation, we have our students, but because we're growing older and the industry is changing so much, it's the most exciting and interesting people that continue to want to be a part of this new age, if you would, in the 21st century of biotech. So it's extremely exciting, and thank you all for being a part of this. We do have some other resources, and I want you to, some of them have been mentioned or referred to. But one of them is something that grew out of demand, so to speak, which is the biotech-careers.org website. And on that website, there are short videos of different careers in biotech. And we've had a lot of high school students that are looking at these different options for themselves, as well as community college, as well as anyone for that matter. The idea behind these videos is that they are there's a variety of different kind of people and a variety of different kind of careers. So it shows the broad spectrum of possible careers within biotech, but it also shows different ages, different ethnicities, all kinds of different people, so people can associate, the students can, or whoever's watching the career video can associate with something that they might not have thought about. We've also mentioned the biolink.org. We've mentioned the different websites for the biomanufacturing, the new website for the Austin AC Squared BioLink Regional Center, and there's just a wealth of information on these sites. We also are very collaborative, and we like to put information and links on each of our sites to these other resources as well. So don't be surprised if you go on the website and you'll see a connection, a link to one of the other centers, because we want to foster that, and we're not in competition with each other. In fact, that's quite the reverse. We really want to work together. There's also links to archived webinars, and that can be found through the Maytech Center and through our website as well. We also have archived materials at ATE Central, and so that's another resource for you. We'd also like you to sign up for our newsletter. You can sign up for, we've cut back on the hard copy newsletter, so we just do those a couple times a year, but we also have the online web, newsletter, and a lot of people are putting information on that. We also encourage you to provide us with articles. If you've got release for a picture means a lot, and we would love to showcase your program as well. We have a couple of really big events coming up. One of them is the annual Summer Fellows Forum, and this year it's June 5th through the 9th in Berkeley, California. We would love to have you come. You can go to the BioLink website and see and apply. We do have some fellowships, and we would love to have you come and also present. So there's on the BioLink website, there's also a place where you can make a suggestion, fill in an application to do a session, to present, to bring materials, to share what you're doing at your community college or technical college or high school or university or company. There's just lots of opportunities for sharing. I also want to alert you to the high-tech conference. This is the High Impact Technological Exchange Conference. This is funded by ATE projects and centers. It started because there is a regular ATE-PI meeting, but not everybody can come to that. It's only for PIs, and so principal investigators are the only ones that are invited. And this conference came out of that, where we felt that it was much more important to also share with those people that aren't PIs, those people that are thinking about writing applications, those people that just want to connect. So this year it's July 17th through 20th at Salt Lake City in Utah. And I want to let you know that BioLink is one of the executive producers. And what that means is that we have paid for 25 registrations, and we give those away. Now, we can't fund everybody's travel, but we can certainly fund up to 25 registrants, and we would love to do that. So with that, we come again to a point where we might want to ask questions, and we've got a couple. When are the Summer Fellows presentation proposals due? The date hasn't been finalized yet, so let's keep watch. And Deborah, you asked a question. I can get back to you and make sure that you know that. Oh, I'm sorry, I missed a slide. We've got some future webinars coming up for BioLink too, and their third-party certification entry-level certificates coming up in March at a date still to be announced, and then coming up a 4-year biotech programs and traditional 4-year colleges that are offering biotech programs. Now we can actually go to the questions. Good. There are some ideas, and I can see where we will have some really exciting possibilities with the Howard Hughes Medical Institute CPhages project for undergraduate research as being one. And so we have thoughts, and we will get that up as soon as possible. And Sandy Porter is going to be part of getting that up on our website, and she's on the call today, so we'll have that up probably by next week. Oh, the application is up now. It's the deadline that the deadline is mentioned, so we'll take care of that. And thank you for asking that. Several people are still typing their questions, but you can see that in the chat box. And I have to say that I personally really appreciate all the presenters today and sharing the kind of information that is helpful for all of us as we think forward. Now, what we've got as we conclude our webinar is a chance to take a survey. So if you will look on your, this is really important to us. This is how we know what we can do to improve, what we can, what we did well. It just helps us a lot as we go forward and create more resources and information to share with you all and to share with each other. Thank you.
Okay, I'm going to launch the survey now, so just give me one moment. As you're concluding the survey, I'll be playing some music, and we would just like to say this officially concludes our webinar for today. We'd like to thank you very much for joining us.